My broader field of research and scholarship is in molecular and evolutionary ecology of mosquitoes, mosquito-borne pathogens. But today I'm going to talk about my work in relationship to resiliency of malaria transmission and the strong vector intervention. Yeah. Okay, so this is the outline of my talk. I will begin with some introductions about what malaria is as a disease. Then I will discuss a little bit about my previous research, my current research, and then I will finish up with a discussion about what my future plans are with research. Just keep on going. Right.
It's okay. Don't worry about it. Don't mind me.
It's not good. When you're done.
Actually, I'm not sure how to do it here in this form. Could everybody online please mute themselves? There's a lot of noise coming through. I know, but I can't. Okay, good.
All right. Hey, what's happening? Okay, for those of you who may not know what malaria is as a disease, malaria is an infectious disease that is caused by infection of the human red blood cells by protozoan parasites in the genus Plasmodium. There are 5 known species of Plasmodium that transmits malaria, of which Plasmodium falciparum and Plasmodium vivax are the most clinically important, and they're also most widespread. These parasites are transmitted by the bites of female mosquitoes in the genus Anopheles. This one. Yeah. Okay, so for malaria transmission to happen, a mosquito must first bite an infected person, picks up the gametocyte stage of the parasite, and then the parasite goes through various stages of asexual development inside the mosquitoes until it reaches the sporozoite stage, so that when the mosquito bites another person, it infects the person with the sporozoites, which first infects the liver cell, and then the red blood cell, goes through a whole complex cycle of a stage development. Some of them differentiate into gametocytes, which are then picked up by other mosquitoes and continue the cycle of transmission. This is a map showing the global endemicity of malaria for Plasmodium falciparum on the top panel, and Plasmodium vivax on the low panel. As you can see, malaria is a tropical disease, so it is therefore found mostly in the tropical regions of the world and affects mostly low and middle income countries, developing countries. There is some variations in the distributions of the species. For example, Plasmodium falciparum in Africa is widespread, whereas Plasmodium vivax is restricted only to the Horn of Africa and in Madagascar. But yeah, and in South Africa, you see a lot of the Plasmodium vivax, but less of Plasmodium falciparum. Okay, so malaria control relies heavily on the use of insecticides to control adult mosquitoes. Okay, and there are 2 common methods in which insecticides are deployed against adult mosquitoes in the malaria community. These are indoor residual spray and long-lasting insecticide-treated nets. The indoor residual spray involves spraying inside surfaces of houses so that when the mosquitoes come in to bite humans during the night, they come in contact with the treated surface and die from the contact with the insecticide. For the insecticide-treated nets, or LLINs, it's the same. The mosquito comes in, get in contact with the nets as they try to bite the people sleeping under it, and they die from exposure to insecticides. But the bed nets itself serves as a barrier too. So even if it's not treated, it protects people from coming in contact with the mosquitoes in the first place. Okay, so beginning around the year 2004 was when there was a global effort to control this disease called Rollback Malaria Initiative. This was spearheaded by World Health Organization, UNICEF, and other bigger agencies including the local programs, the national and the subnational programs, malaria control programs in the malaria endemic countries. So now that is not to say that there are not other methods of controlling mosquitoes. We have various others, larval control, adult control. For example, larval control includes source reduction, chemical larviciding, biological control. Adult control also has methods in which you can use. But in the malaria world, it's mostly what I just described in the previous slide. And there's tendency to move towards, much more newer technology for control. This plot shows the global malaria trend from the year 2000 to the year 2019. On the y-axis is the incidence rate, which is the number of cases per 1,000 population at risk. As you can see, this was when the global vector initiative, which I talked about earlier, was implemented around here. And you can see there's a downward trend in malaria burden or malaria incidence up until around the year 2015 was when we begin to see a malaria reduction begin to stall. Despite more efforts to distribute bed nets and insecticide spray, there was no further gains beyond that. So that became an interesting question for which the scientific community began to realize that malaria transmission has become resilient to vector control globally. Okay. Now, my previous research focuses on evaluating the impact of bed nets, LLINs, in Papua New Guinea, which is my country. From now on, I'm going to use PNG to abbreviate it like United States, US, for example. Yeah. This is where Papua New Guinea is on the globe. For those of you who may not be familiar with, it is to the north of Australia, east of Indonesia. These are my study sites. So my study was based in a province called Medang. It's a malaria endemic, very holoendemic. Okay, and my samples were collected from these 6 villages from the coast of Medang. All right. In PNG, the main vector control intervention method is LLINs, bed nets, no IRS. We didn't do any indoor residual spray. There is talk currently going on about it, but we have not had any history of IRS. Long before that, but not recently, since the rollback malaria initiative we done. This is similar to what I showed about the global malaria trend. This is for PNG, okay, from the year 2000 to 2015. This was around the time when LLIN was distributed. Now you may ask why there is a downward trend before that. That's because people were already accessing bed net long before the program came into effect. Okay, so there's a downward trend all the way to 2015. So you can see where malaria has been going. Okay, now one of the metrics that I used to evaluate the impact of LLINs on malaria in my country was to look at the human infection prevalence in the country. And what we found is that in 2017 was the data I myself collected this year here. And I compared the results with the data collected by my other colleagues from year 2006 to 2014. And as you can see, for Plasmodium falciparum and Plasmodium vivax, the prevalence of infection was above 40% at the time. And when the bed net was introduced around the year 2009, after that, you see a dramatic decline, all the way down to below 10% for Plasmodium falciparum in the year 2014. However, in 2017, in 2017, you see a resurgence, a rebound in the infection prevalences. Plasmodium vivax has it early 2014. Okay. The other metrics that I used to quantify to measure, to evaluate the impact of vector control to malaria was transmission rates. Now, this is measured in terms of entomological inoculation rate, which is defined as the average number of infective mosquito bites that a patient receives in a year. Okay, and it's quantified by taking the product of these 3 quantities. Okay, B is average number of mosquito bites that a patient receives in a night. S is the proportion of all the tested mosquitoes that were positive for the sporozoite stage of the Plasmodium parasite. And 365 is the number of days in a year. You do that, you get your EIR. Now you can measure you can look at the transmission. Anyway, in order to get such data, you have to have good methodology to collect mosquitoes. Yes. So this is not me. This is one of the village collectors who uses the human landing catch method to collect mosquitoes that land on the body during the night, and then puts them into cups that are labeled according to the hours of the night in which they are collected. That is used to measure the human biting rate per night, as I discussed earlier. And those mosquitoes were then brought back to the lab, and we ran PCR to test for the species of the mosquitoes, and also the qPCR to detect for Plasmodium sporozoites in the head and thorax of the mosquitoes, so that we can calculate the sporozoite rates. Right. So then here is the result. This was my data for 2017 compared to data we collected before that. Again, BedNet was introduced in 2009. Same story with the infection prevalence. You see a rebound in the transmission rates as well. Okay. So there's consistency between these two metrics. Now, in the scientific community, at least, in the malaria research community, there is an understanding of what we now call the transmission reduction resurgence phenomenon. Okay, from the time you introduced a vector intervention method, you see a dramatic decline immediately after that. However, it reaches a certain point below which no further reduction can be observed. And that persists for quite a while, for some time, until you begin to see a rebound. And so the research communities believe that this is due to pre-existing and emergent vector resilience factors. Okay, so the natural question that comes to your mind is, what are some of those factors? So here is a list of the factors that I did my study on to try and see if these are the main or some of the factors that are contributing to the resurgence in the PNG, in the context of PNG malaria. Okay, these are insecticide resistance, biting behaviors that reduce the risk of contact with the LLINs, plasticity of host selection, which allows the vectors to evade the bed net, and also aggregated human vector contact, which I will explain a little about that later on, which sustains transmission in the presence of interventions. Okay. So the first three are the subject of my previous work, and the fourth is the subject of my current work. Now, when I first started, the first thing I did was, one of the first research I did was insecticide resistant surveillance. And my work, which was published in 2010, found that the mosquitoes, the natural population in PNG, were 100% susceptible to the insecticides that were used to treat the bed nets based on WHO bioassay tests. And this is a graph showing the survival curves for our test. This is the proportion of mosquitoes that survive, and this is the time after they were exposed to the, and you see by about 30 minutes after, 40 minutes after they were all dead. And up to the current day, as recent as 2020, the work by my other colleagues continue to find 100% susceptibility in the populations there. One other factor that may become, it's probably one of the main factors, is outdoor biting. So the mosquitoes come into the village during the night. They are nocturnal biters, as you know. So they come in during the night, but they bite outside houses because the bed nets are deployed inside houses. And so they are not coming in contact with the nets, which are deployed indoors, and the transmission is going on from outdoor biting, right? So what we found in those villages, where the number of bites were the same in indoor and outdoor. So there is 50% transmission going on, which might explain the persistency despite, intense vector intervention. The other factor is early biting. The mosquitoes come in earlier when people are still awake. They're active. They're not really tucked under their bed nets. So a lot of transmission is happening long before they get under the bed nets and assume protection, right? And we also find that about 30 to 50% of the mosquito bites took place in the evening, right? That's a lot of bites. The third factor is plasticity of host selection. And I'm going to explain this a little. This is defined as the ability of the vectors to utilize any available vertebrate host species within the foraging area of the mosquitoes, rather than having a fixation on a particular host species, for example, humans. So if they're fixated on human, this is anthropophagy. Okay, you call that the tendency to prefer human blood more so than other animals. So, but if you are flexible and you can bite any host, that's plasticity or opportunistic. Okay. The relevance of this kind of behavior to vector control is that because LLINs is an indoor focused intervention, and LLINs therefore targets anthropophagic vectors because they are more likely to pursue humans indoors and therefore get in contact with the nets, whereas it has less impact on the opportunistic ones, their population. And when I'm talking impact, I'm talking about the mosquito population, not the individual. Okay, and that allows for the transmission to persist because of the different strains or different behavioral subgroups within the population. And this evolutionary implication to this behavior is that LLINs can change the vector population structure by selecting for those individuals with the opportunistic traits over the anthropophagic ones. And in doing so, it becomes self-defeating for the LLINs. It's there to control the mosquitoes, but in fact, it's actually changing it so that the mosquito is resilient to the LLINs. So, now again, to study plasticity, you have to have a good sampling method. One of the highlights of my work was to develop a method that is suitable for sampling blood-fed mosquitoes in PNG, which is different from African mosquitoes, which are mostly anthropophilic. They rest indoors, sorry, endophilic, because they rest indoors. Whereas in PNG, they are mostly exophilic. They're outdoors. They come and bite, and then they go and rest in the vegetation. So you have to have some kind of a sampling method to do it. So this is called a barrier screen sampling method. It's this structure. This is garden shade cloth. You just set it up like that at the edge of a village between the bush and the village, so that during the night, when the mosquito come to the village to feed on the domestic host and humans, and when they exit of the blood feeding, they rest on those surfaces of the screen, and you have a collector comes along and collect it with the aid of a aspirator and a handheld flashlight. Okay. Then earlier on, when I was doing this study, we didn't know the host range for the malaria vectors there. You know, what are they feeding on apart from humans? So we use the metabarcoding analysis, which is basic genomics. Okay, extract the DNA from the mosquitoes, run some PCR with 16S, which is that amplifies any vertebrate hosts, and then do sequencing of these PCR products, and then do some bioinformatics to try and match those different sequences to the hosts that are available in the database, public database. And then we found out that the host range for PNG vectors is pretty narrow. 98% of the mosquitoes were biting on humans, pigs, and dogs. But we also find 2% of those mosquitoes were biting on fruit bats, also on some mice, mouse mice, and also on opossums. Yeah, so there is that. Now we know that these mosquitoes are biting mostly on humans, pigs, and dogs. Why would you do a lot of genomics? And so we developed a qPCR methods that with primers and probes that targets the 3 hosts, and then we use that cheaply to routinely analyze the blood meals from thousands of mosquitoes that are thrown at us by the field technicians. Yeah. And then we use the blood meal data to quantify the host selection from all this method. Now, we quantify it in terms of theta statistic, which is simply the proportion of the mosquitoes that fed on humans divided by proportion of humans in the proportion of available hosts in the foraging area, in the community that were humans. It's like a ratio of ratio, like an odds ratio. Now, if your theta statistics is equal to 1, that means that both humans and animals were utilized in proportion to their relative abundance. If, however, your theta is greater than 1, that means humans were overutilized and animals were neglected or underutilized. If your theta is less than 1, that means humans were underutilized, even though they may be in abundance. Animals, however, had a higher, they were utilized more. So there is the opportunism, anthropophagy, and zoophagic tendency based on the statistic that comes out of your data. And so this graph shows 3 different species of Anopheles vectors for malaria in PNG, and the different populations for which we collected the mosquitoes to do the analysis. And this is the theta of 0, and the open circle is the theta statistic that you're comparing it to the threshold, to the standard, to 1 with the confidence interval band. As you can see, there's variation among population in terms of which way they are feeding. Down here, they're feeding mostly on animals. Above the 1, they're feeding mostly on humans. And if the confidence interval crosses the dotted line, that means they are feeding in proportion to the relative abundance. So there is, because of this, if the mosquitoes were mostly anthropophagic, you would see all this would be above the 1. But there is fluctuation, which means that these mosquitoes exhibit plasticity of host selection. That gives them the tendency to, like, if the bed net pushes them away from humans, yeah, their population can still survive by feeding on the non-human host, and also feeding on outdoor and early biting sustains transmission. To combine all of these factors together. Now, the summary of my previous research findings is that malaria transmission in PNG has rebounded in recent years, and that outdoor biting, early biting, and plasticity of host selections are likely factors that underlie the phenomenon of persistent transmission or resilient transmission that we see in PNG, and you can almost generalize these to other parts of the world where malaria is a problem. Okay, and the implication of this finding is that reliance on the LLINs alone is not sufficient to control vectors, and therefore alternative vector control methods, including novel ones, must be explored and implemented alongside the existing method. And thirdly, an in-depth understanding of local vector ecology is critical at any stage of vector intervention program. And you would agree with that as scientists, I'm sure. So that was my previous research, and now I want to switch to talk about my current research. Okay, my current research is focusing on understanding the relationship between the phenomenon of aggregated human vector contact and malaria transmission in Kenya. And I'll explain that in my next slide. Now, you might wonder the transition. I think most of you have already know. Before that, I was working in my home country, and when I transitioned to UCI for my postdoc, my advisor's research was based in Kenya, so I had to bring my ideas and go along with him. So that's why. This is Kenya on the map of Africa, for those of you who may not be too familiar with where it is located. This is the malaria trend in Kenya. On the y-axis, again, this is the malaria incidence, and you can see that the bed nets and IRS and bed nets were introduced in Africa, okay? It's not just one, both. In some cases, they are one, but mostly it's both. Around 2006, and you see a downward trend, but now going back up in recent years. Okay, now let's talk about aggregated biting. This is a situation where when the mosquitoes come into the village to bite a village or community, a few individuals they receive a high rate, excessively high rate of bites, whereas the rest of the community receives a low biting rate. So that creates some kind of a binomial distribution pattern in which most people receive 0 bites or 2 or 3 bites, while few individuals get a higher number of mosquitoes getting in contact with them. Now, before I explain the relevance of that phenomenon, I want you to understand the basic reproduction number concept. It's an epidemiological concept, and it basically means that there's a primary case, someone who is infected, and anybody else that becomes infected as a result of transmission from this person, the number of people is called basic reproduction number. For example, if 4 people get infected, then the R naught, which is basic reproduction number, is 4. Okay. Now that you know that, let's talk about aggregated biting. What we have seen, the models, and also we have fitted empirical data to the model, have shown that as the level of biting aggregation increases, so does R naught, with transmission rates increases with the level of aggregation. Now, interestingly, we don't know what the mechanism is that causes this relationship at the first place. We see it, we don't know what it is, what's causing that. So my hypothesis is that the transmission is structured in such a way that you have this subpopulation of humans, few individuals with high rate of bite, and then you have the rest of the community with low biting rate. So malaria transmission is actually maintained intensely between the subpopulation and the mosquito population here. And then the rest of the community becomes infected from mosquito bite that spills over into the, so then you can think of this group as the primary case and those groups as the secondary case. Because if these people are constantly infected, you think of it in a population level, not individual as I described earlier. So these people are less like they get infected and then they lose it. So they lose immunity, but then, and so they're susceptible again. So it keeps that from going. So that explains the relationship before. Okay. So this hypothesis forms the basis for an NIH grant proposal, it's an R21, which I just submitted 3 weeks ago today, before I came here. And these have two specific aims. Aim one is to delineate the human subpopulations that vary in the rate of mosquito bites and the factors that cause that pattern. And secondly, to determine the contributions of these different human subpopulations to the transmission of the parasites into the vector population. Which group are the drivers of transmission? Because they could play a role in the concept that I painted earlier. There are two study approaches that I'm using to analyze the two aims. The first aim will be using microsatellite multilocus genotyping, which is DNA fingerprinting, which she touched on. So what we do is we collect all those blood-fed mosquitoes, right? And then test them if they are humans using qPCR, and if they are, then we apply, we test for the genetic fingerprint in the mosquito and match it back to the humans in the village from which they fed. So that way we can match the mosquitoes and know which people in the community are actually contributing the most to mosquito blood meals. Which also tells you who is being bitten more and who's being bitten less. The other thing is to look to link infected mosquitoes back to the source, human source of infection by matching the parasite haplotypes. Okay, so you do that here, this grid matches this person. So in that way, you can tell the members of the community that contributes the parasite flow into the vector population, and are they consistent with the people up here that we determine. That's the goal. So I have done some preliminary work, and I'm still doing that. And if I come here, I will continue to do that. You know, yeah. So these are my field crew in Kenya. They're collecting the well-trained shopper. So we collect mosquitoes using different methods. This guy is collecting human blood samples, and then we bring the samples back to the lab, and we run them through various assays using quantitative PCR to identify Anopheles species, identification of the blood meals to determine if they fed on cows. Cows is a big thing in Kenya, humans or whatever, and then detection of the parasites, and also do the two methods that I described earlier on in my talk. Now, these are 3 students who are working with me right now. They are well trained to carry out all of these approach. So if I come here, if any student is interested to work in my lab, you will be welcome. Just let me know. Yeah. So this is some preliminary data. As you can see here, this on the y-axis is the number of different humans that receives the different number of blood meals. Okay. So we see that a lot of people, 84 people, 68% contributed 0 blood meals in the sample. We didn't match them to any of those mosquitoes. However, 1.6%, 2 people contributed to 50% of the mosquito blood samples. Now you may ask, is your sampling biased? No. We made sure that the sampling was even, such that there is no human error in this. So this tells us exactly highly aggregated biting happening in the community. Okay, so now, so from that data, we then categorize the people who we had the genetic fingerprints into 3 groups: those that had 0 contribution to blood meals, those that had 1 to 5 blood meals that were matched to them, and those that had greater than 5 blood meals that were matched to them. And then we look at the age categories within each group. And what we found out for these two high mosquito biting group is that school-age children, 6 to 15 years old, constitute the largest proportion, okay? The green one, the largest proportion of those, but they're less so in here. Okay, and also the infection prevalence is high in this group as well, as you can see on the graph on the right, on the left, your right, my left. Yeah. So it's can you then conclude that this group are school-age children, or at this point you're leaning to say that because that's what the data is showing, but these are preliminary data, and there's similar analysis like that I haven't done yet, but a lot will be revealed through this kind of work. This is just one test, a lot more analysis to be done. So now the relevance of this kind of work to resilience of transmission is that vector intervention reduces biting rates generally, but in doing so, it promotes the phenomenon, it promotes aggregation. Think about rivers, yeah? When you go to, you see a lake and there's a water level, right? You don't see the rocks, but when the level drops, the rocks begins to surface. So if you push transmission down, then you cause aggregation, yeah? And so then it forms a resiliency again to the control program. So therefore, delineation of those human subpopulation is important for vector targeted interventions. Targeted intervention is a method or is an approach for which you distribute or deploy bed nets and antimalarial drugs to a subpopulation, okay, that are considered risk group or super spreaders with the goal of reducing the transmission community-wide. And this is important for resource-limited countries. Yeah. Now, I want to finish with my future plan. So here at Rutgers, I would like to develop a research and training program that under the theme molecular and evolutionary ecology of mosquitoes and mosquito-borne pathogens. I want to do two research arms. One would be local program and the other is international program. The local program focuses on arboviruses, in particular Eastern equine encephalitis and West Nile virus. The international program focuses, Jamestown Canyon as well, international program for malaria. And I'm thinking of for the local, both vector ecology, evolution and control, as well as virus transmission, emergence, surveillance, all of that. The study sites include here in New Jersey, the northeastern region, and I also want to continue my Midwestern region connections with people from there, Michigan State especially. And I'm thinking the main collaborators are, of course, the faculty members here at Rutgers, the Center for Vector Biology here, the New Jersey Mosquito Control Agencies. I look forward to work with you guys on that and various others. For the international component, again, Anopheles ecology, evolution and control, as well as parasite epidemiology and transmission. And my study sites will be based in PNZ and Kenya. Okay, and my collaborators are in place. They are there. We work together all along. I'm just gonna, switch the institution here to Rutgers. So, okay, so I've done some work with respect to JCV transmission in Michigan, and what we see is that those mosquitoes that have a very low abundance tend to have high infection rates in the mosquitoes, and in particular Anopheles communis and Anopheles Aedes communis and Anopheles punctipenis. Yeah. Okay, so I have talked about my grand vision, but these are the projects that I hope to work on in the short term for the next 5 years when I come here. Okay, so this one is just an extension of that. The first one is what I have described earlier, which is my current research project, and then several others that I'm working on. They are either in the writing stage or in the development, or one have already been submitted. So we can talk about that. And of course, I'm happy to be part of the teaching program here. These are a list of courses that I can teach in addition to whatever the department can assign to me for that. And I would be involved in the graduate mentoring and undergraduate mentoring as well. Of course, I will be happy to be part of providing services within the department and also be involved in the extension programs that you guys run. Yeah. Yes, and so My career has been I got here by the support of various individuals who helped me. Yes, in particular, my former PhD advisor and my 1-year postdoctoral advisor as well, my current postdoctoral advisor, and the various people who helped mentored me and supported me throughout my career. With that, thank you for your attendance, and I'm happy to take any questions.
With the aggregated biting
Okay.
behavior, so the answer to my question is the school age children, but are there certain behaviors that you've been able to identify that lead to this aggregating biting behavior of people or of the mosquitoes?
Could you please repeat the question for the audience online?
So the question was, is there any behavior in the school, the school age children, or any people in general?
In people in general that
Okay.
lead to this aggregated biting.
So we have yet to analyze those data, but I think one of the environmental factor could be behavioral, like people spending more time outdoors rather than going to sleep early. Maybe those are the ones that, or that they may not have bed nets, or they refuse to sleep under bed nets. That happens to receive the most number of bites. Or it could be that their communities are positioned close to the larval habitats, for which the mosquitoes can easily commute, bite, go lay eggs, and then live in a neighboring community, yeah. So there are many factors, and certainly behavioral components will be evaluated.
Follow one of this, is there a sex bias? Do you see?
Yes, yes. Because of time, I didn't put that slide up, and I have it. So the males have higher biting than female counterpart. That's a very interesting observation. Also, that the infection rate is higher in males than females. So good question. Go ahead.
So you also talk about the data, so data, some
Theta.
calculation,
Statistic, yeah.
theta, whatever.
Mhm.
So it tells if the human or the animal is overused.
Yes.
Right? It seems that the quality or the group of people is more important than the number of people. Like you talk about the aggregate that they biting. So it's not the number matter. It's so the special groups are more important than the number of people. Right?
So his question is about the relationship between the theta statistic that I described earlier and the aggregated biting. Is that right?
Yeah.
Okay. So these are two different things. So the theta statistic measures the host selection, the tendency to bite humans or non-human hosts, whereas aggregation is only for the humans, within humans alone. Forget about the other animals. So within humans, which people are bitten the most? Yeah.
I was thinking, because you present the data on aggregation, right, and you see children and young adults to be the groups that are affected the most. But I mean, the behavior of the mosquitoes in Kenya, they are endophilic, right? So
Yes.
whatever is happening in the house, and that might be have to do with behavioral in terms of protection with nets or
That's
use of them.
yes, those are possible. So we have data, and we have designed a study design in, sorry.
So if that's the case, is that in a group of houses where the children don't live, or is a behavioral thing linked to people that don't have children, or?
So one thing we think is that the school age children tend to like they go to boarding schools, yeah. And so when they come home, they don't really have like a setup in which they get protected by like bed nets available to them to sleep under. So that is one thing we're thinking. This data guides us, like maybe that is one factor. So they go to boarding school and then they come home and they don't have like the bed nets there to sleep on, and they're exposed because they're not going to stay for a long. They come and go. And this study was was done during that period when, yeah. So.
Yes, I'm interested in when you did collections, mosquito collections for the aggregation study, how did you reduce sampling bias?
Good question. So we have 40 houses, and then we made sure that every houses were sampled with the same effort. Okay, same number of days, same, if you're using 2 Procopex to go in there and get mosquito, you stick with 2. In this case, we use 1 Procopex only, and we also use clay pots to sample outdoor mosquito outside the house. Yes. Yeah. So we made sure that there is a balance across 40 houses, so that if there is any bias, it's to do with the different houses variation, and not because we've sampled more in one house and less in the other house. Yeah.
So you had an equation with the EIR where part of it is the bites per night. But then you also talk about how a lot of bites happen earlier in the evening. When you talk about bites per night, is it like that the whole entire time, or is it only when you're done and sleeping?
You can wait. Oh. So you can partition your your analysis to whatever time of the night, depending if you're trying to estimate transmission in the early evening, you sample mosquitoes in the evening.
Okay.
Yeah. Or if you wanna, but in this case, you have for the overall picture. If you want to get an idea of the trend generally, you do all night sample. Yeah. So that captures every state.
For the average biting, is there any difference among the blood types?
Good question.
And also.
Could you repeat the question?
Oh, yes.
The
Good.
second one is, so is there any difference among, if you eat different food, is there any difference about the biting?
The food?
As I remember when I was young, my dad said when when he drinks some bite weed and he couldn't feel like the mosquito doesn't bite him. I don't know if the mosquito really don't bite him or he just couldn't feel anything because he drinks.
Yeah. So the question was, is there any variation in the biting rates among humans who have different blood groups, blood types, and also if they have the variations according to meals, the kind of food they eat. Okay, so the question to answer is blood types. Some people have done some studies on that. They have some. Becker did some. Yeah.
You're less likely to be dead if you're A.
Yeah, A group. Yeah. So there is some data that shows that, although I have two
papers.
Yeah, there were 2 papers on it, and that's it. Nobody followed up with it. But it will be included because we are collecting the blood samples anyway. So all of this will be analyzed and tested as part of the study. But with your question about food, that would be, let's say, this when I listed all those projects that will be done in the future that I plan to do. One of them, the second one, let me see if I can get there. Oh, sorry. There you go. Yeah. I think that the functional genomics would address some of that. So what are some factors in humans that affect the mosquito behavior so that they aggregate on them, right? And so maybe that you can do some metabolites, maybe that you can trace it back to the kind of food they eat, so.
I have a question. Oh. I have a question about the conformability study when you're talking about how the use of LLMs is actually making mosquitoes choose other times of the day to bite or the night to bite, but also making them more opportunistic in biting other animals. Because one thing you didn't cover was the fact that no other animals can maintain human malaria parasites. So a mosquito that is biting a pig or a non-human organism is actually, it's a wasted product from the point of view of the mosquito. So that technically that would decrease the strength of transmission.
That's true. So you want me
to? It's a good thing.
Yes, it is. So
but why is it going up?
let me explain. So her question is about if the mosquitoes are biting the non-human hosts, which are not hosts for the parasites, then it should be diluting the transmission. It should reduce transmission. It's a good thing. And there is this whole vector intervention method which involves zooprophylaxis. Yeah, the idea of pushing the mosquitoes away from humans so they can feed on the animals, which then adds your question. So the concept about so there is the zoophagic mosquitoes who like to zoophilic mosquitoes, zoophilic, who they like entirely to feed on mosquitoes. Non-human hosts. Those are the kind of mosquitoes that are, they're not vectors, they're not good vectors. But the opportunistic ones, their population is sustained because they are feeding on the non-humans. They have enough mosquitoes to go around. A lot of them, high population size, excess mosquitoes. So now you have people behavior plays into it. They're biting outdoors. Because the goal of IRS, for example, is to get the mosquito numbers down,
Right.
down, so that it minimizes the transmission, because transmission increases when vector abundance.
The non-human animals are maintaining the population.
Exactly.
Maintaining them high.
Exactly. They make up for it. And then by chance, they bite humans, which is why you can push malaria down with bed nets, but it comes to a certain level where it's,
So follow-up question, which is, so this thing about acquired immunity. You get
Acquired.
enough bites,
Mhm.
and if you survive it as a child, you then become basically resilient. You don't get sick. You're still a host.
Mhm.
So is it what happened is that the efforts brought the transmission low enough
Good point.
but the measurements you had was actually infection, it wasn't disease. You're looking at actual
Infection,
rates, right?
yes.
So tell me more about that. What is,
Yes.
are you seeing the sort of this, and actually that goes too with the secondary, you have these primary hosts,
Mhm.
which it was interesting you found were children, because they don't have yet the opportunity of developing acquired immunity. So this secondary set of hosts, are you seeing more disease in adults? Because they only get bit every once in a while?
The infection prevalence is high in most age group. There's some variation, there's variation really, but they're high enough.
So the EI.
Yeah.
Whatever the
EIR?
population is really high, is enough for resulting in acquired immunity across the board? In Papua New Guinea, do you see acquired immunity in Papua New Guinea?
Yes.
Okay.
So the adults mostly they have this immunity, but there is also the problem of if you push the transmission down so much, people lose immunity, they become susceptible, and now you see an epidemic. And then, there is that concept. But there's a lot of asymptomatic infection going on as well.
Right.
So now this is the epidemiologists are looking at it. Yes, but it's a very interesting question though.
Because it
Yeah.
affects this model of,
Yeah.
of hot spots of transmission. Because if you have a population that is primarily, like the way I think about Kenya,
Mhm.
is that you have all these adults that I always thought there was kind of an even likelihood of getting a very high equal likelihood of being bit by an infected Anopheles gambli or
Yes.
one of those. And so therefore you have this system where you have these kids, if they survive, then they become asymptomatic.
True.
And I was curious to know, in Papua New Guinea, you had way more mosquitoes.
Yes.
And you had a tremendous amount of variability in their anthropophagy. Some of
Mm-hmm.
them were really very zoonotic, and then you had that Farauti that is
Yeah.
kind of the primary anthropophagy.
So things have changed a lot, but in the past, the likelihood of a child surviving into adulthood is low. Back long time ago. So because he would die from malaria. It was one of the factors that determined the human genetics in the country, But now it's okay. You have healthcare, so people can live long now. But the question still remains as to that there is enough transmission to maintain immunity, in the population, right. But the transmission is to do with human mosquito interactions, but the immunity is one factor that might regulate a little bit about where the parasites may be mostly found. But the phenomenon of asymptomatic infection makes it now that people can walk around looking healthy and yet carry the infection in them, and that contributes to the bites going around.
But this kind of immunity, you said acquired, what is it called acquired? Immunity is acquired.
Yeah, it is acquired.
I remember it was called pre-immunity.
Oh,
But anyway, Doesn't that also suppress the number of parasites you have in your body, but that's the likelihood of a mosquito picking it up?
It does. It does, but like I said, there is somehow, and I have yet to study this in how the physiology of the mosquito, of the parasite in the humans with immunity, you know. So did they find a balance in which they can coexist for asymptomatic ones, for example? You know, it's not causing them to be sick. Maybe it's low parasitemia.
Yeah, it is
Low parasitemia.
it's low parasitemia, which may make some mosquitoes non-vectors, but the good vectors are still gonna.
Still good.
Yeah, sorry. So this is a question that jumps off of your slide about the variation you saw between different sites in the proportion of human blood meals you found.
Okay.
I'm just curious if the mosquitoes actually are evolving, or like the actual preference for humans is evolving, or if it's just kind of like utter happenstance. That if you took those mosquitoes and put them somewhere with no bed nest, they would still go right for humans, even if they were biting animals previously. So do you have any gut feeling? This is an annoying question. Do you have any gut feeling about whether that you're seeing an evolved change in the preference? And I guess the things that would inform this would be, you know, are these places where there's tons of bed net coverage? And secondarily, are people keeping animals close to home?
Mhm.
You know, like where I work, people keep their pigs and cattle very close nearby because they don't want to lose the wealth if you know, to cattle or so.
Okay.
Do you have any of that info to kind of inform whether or not it might be a true evolved preference difference, or if it's just kind of the bed nets are doing what the bed nets are supposed to do?
So the question is about whether or not bed net is changing the host feeding patterns, host feeding traits in mosquitoes. Now the answer to your question is like that is one of the area in which we want to investigate. That's what I want to study that, which is that is there like different strains of vectors in the population that some tend to prefer humans, others prefer non-humans, such that that's what I meant by evolutionary implications, is that when the vectors, the bed net then, because of that, change the population structure and kills all the anthropophagic ones and then selects for the dominance of the anthropophagic as well, the opportunistic vectors. Now that is under study, and it is one of the areas for which I hope to like maybe we can fill out.
Yeah, sure, sounds good. You still need to do a proper like olfactometer.
Yeah,
You know,
there's a lot of what going on in
it's hard, but.
Yeah, there's those people who does work on that, but that doesn't capture what happens in the field. I think that, okay, if you force the mosquitoes to go one way or the other, they might, but if you give them the real environment conditions, then you can learn a lot from them. But I do think, in my mind, I think they are substance that can different. So we'll see. Yes.
The human genotyping for blood meals is really interesting. But my question is, for that to work, you kind of have to be able to genotype the whole population. Do you have any trouble getting people to participate, and are you able to genotype the entire village?
So the question was about the ability to sample enough human or the rest of the populations in order to genotype to get the DNA fingerprints so that we can match it back to the mosquito. And is it feasible, right? So, like in a place like PNZ, that's one of the reasons why I'm doing this work in Kenya, is that it becomes really difficult because the mosquitoes are biting, they're coming in and biting and then they're exiting. So it's very hard to do it unbiased, to sample the mosquitoes in an unbiased way, which means you have to sample the entire community, right? And that becomes a little challenging. Yeah. Only those who volunteer would do, but in Kenya, we do it by household. We do it by household, so that is a representative example, and you just, because you're collecting the mosquitoes that fed on people within the house, and if you have enough houses, and they are all, and so far they have all been, when at Kenya they gave everybody were okay, so we took their blood. Consent.
Good IRB.
Yeah.
So the mosquitoes that you collect from inside the house are guaranteed to have fed on the people in that house?
No, they could have come from a nearby, but we would exclude that from the analysis, yeah. Because they would have beaten someone next door and moved here. Or, yeah. So we would exclude that because they are not in the humans are not, we don't have the. You will own it, but the mosquito will collect it in this house, that high likelihood of biting it in this.
Yeah.
So infection with the parasite changes the mosquito behavior and they bite more. So are we assuming that infected mosquitoes are distributed randomly throughout all of these populations, or is it possible that these folks that are being bitten aggregately are just interacting non-randomly with infected mosquitoes?
So the question is whether or not infections, human infection status, like having gametocytemia or so, is a factor that contributes to the aggregated biting, so that mosquitoes will bite those people. That a parasite affects the human physiology that makes them susceptible to higher rate of
Affects the
bite.
mosquito behavior.
Yes, yes. Yeah, that's right. So that is it's an unanswered question. It's one of those areas of investigation. And I've had some hypotheses about that perhaps those individuals with a high, and if you think about it in terms of evolutionary basis of such thinking, it would be beneficial for the parasites too. So there could be some coevolution in a way that the parasites are producing a lot of immunity. In the humans, but I don't know how it I don't know how it might affect the mosquito effect the mosquito to don't come bite on those people. It could be like producing some kind of even body immunizations.
Well, I mean, it's actually kind of complicated. But
Yeah,
I mean,
it's very
there's evidence that the mosquito